The overall goal of this procedure is to trap micron-sized biological objects with plasmonic and photonic crystal tweezers. This is accomplished by first fabricating the plasmonic substrate or photonic crystal substrate. Next, the biological sample is prepared by adding BSA to the cell solution and providing adequate spacing for the cell solution beneath the top cover slip. Then, the laser is aligned to the microscope, which has been fitted with the correct filter scheme. The microscope is now focused on the sample, and trapping is performed. Finally, micro or nano-sized particles can be trapped with reduced incident intensity through the aid of plasmonic or photonic crystal nanostructures.
The main advantage of this technique over existing methods, like conventional optical tweezers, is that conversion of the optical fields through plasmonic and photonic crystal nanostructures reduces the intensity required to trap small particles, like cells. And it also doesn't require a tightly focused beam, which reduces the risk of cell photo damage from exposure to radiation. This method can help answer key questions in many biological fields, such as cell-to-cell interaction and the study of various forces in biology. It can also be a useful tool for fabrication in micro and nanotechnology by providing precise manipulation of micro and nanoparticles.
To form a gold nanoparticles array using an e-beam evaporator and chromium as the adhesion layer, evaporate gold on a glass cover slip to a thickness of 20 nanometers. Clean the sample using oxygen plasma for about 1 minute to remove organic impurities on the surface. After inducing the polystyrene monolayer to self-assemble, dropcast the liquid onto the